organizing and for the lovely chocolates that showed up at my door this morning. It's really a pleasure to be invited, and I'm looking forward to telling you guys a bit about what we've been doing in our project with bacteriophages, and thinking about bacteriophages and how they can help the honeybee industry, and particularly with the problem of American foul brood. So I'm going to share my screen so that you can see my talk. So the project that I'm talking to you about today is the ABATE project. And the ABATE project is something that we started maybe 3 years ago. And now we're looking to expand the project and kind of open it up into some new directions. And it's really all about using bacteriophages to produce, to further support the health of hives. So first, I'd like to start by just saying that, you know, the work that I'm talking about today is primarily really done by the brave postgraduate students in my laboratory at Massey University. So I'll point out the students that did the work as we go. And also, I need to say right off the bat that without the support of the Howard Hughes Medical Institute's training and education project, we really wouldn't have started the Massey Phage Fauno, and which is the group of faculty, phages, and students that are involved in phage hunting at Massey University. And we've gotten a lot of really generous support, both from AgMart and from the Sustainable Farming Fund. And I should mention also the New Zealand Honey Trust most recently. So we've had a lot of support, and there are a lot of other people that are involved in this work. So the first thing that I'd like to do is just introduce you to a bacteriophage. And I'll show you some real photographs of bacteriophages later, but basically this is what bacteriophages look like for the most part. So they have a capsid that's made out of protein, and they have a tail, and they have these little connectors, which are part of their tail that allow them to identify the bacteria that they infect. And they're incredibly numerous and absolutely everywhere. So you can find bacteriophages in the milk in your refrigerator, in your honey, in your skin, in your gut, in the soil around us, in the oceans. Bacteriophages are 10 times as numerous as bacteria, which are already incredibly numerous. So they're extremely common, and they are everywhere. And it's been over 106 years since we first discovered these bacteriophages, which really means kind of eaters of bacteria. And so what do they do for a living? So a bacteriophage is really a protein particle that floats around in its environment, kind of bumps into things. And as it does that, if it recognizes its specific host, it is able to inject its DNA into that host. And the DNA is able to take over the replication machinery inside the cell and make as many as between 10 up to 200 copies of that bacteriophage in as little as 20 minutes. Once those are assembled and inside the cell, they burst out of the cell. And when they do that, they destroy that bacterial cell. And this is the way that they commonly operate, and it's called a lytic cycle or lysis. So then each one of those individual bacteriophages is able to go on and infect more of that same type of cell. And so when this was first discovered, it was actually before we discovered the incredible power of antibiotics. And because of that, today, if you take an antibiotic like meropenem and you put it in droplets on a bacterial lawn, on a plate, on an agar plate, you can see clearings, and you can see this is the evidence that bacteria are being killed by that antibiotic. And similarly with bacteriophages, once you find them, if you drip them onto a similar type of agar plate, you actually can see that the bacteriophages kill the bacteria. So somebody has just asked, and I'm kind of keeping my eye on the Q&A, but I might miss some of these. So a bacteriophage is a virus. A bacteriophage is a virus that specifically infects bacteria instead of infecting something else like us or bats or anything else that you might be thinking about, particularly in our coronavirus age. So these are viruses, and they're everywhere, and they kill bacteria. All right, and because they have this power to kill bacteria, they've actually been used as medicine in Georgia and Poland and Russia. And I've actually visited a bacteriophage factory in Poland where they were producing bacteriophages for industries like the poultry industry to kill bacterial pathogens in poultry. So this is a tool that's being used in some parts of the world, but it hasn't had a lot of use until quite recently in Western medicine. But very recently, we've seen the rise of stories like Stephanie Strathy's story of bacteriophages being used to actually save her husband's life when he was infected with Acinetobacter baumannii. So this is a recent book. I think it came out 2 years ago, where Stephanie tells the story of what happened to her husband and how American scientists that had discovered bacteriophages were able to take those bacteriophages and administer them to her husband, Tom, and actually save his life. It's a great story. This is also, bacteriophages have also been approved for use on food. And here in New Zealand, we even have one phage product that's used in the beef industry in order to destroy E. coli. So these are not incredibly, not used all the time, and something that might be a little bit unfamiliar, but they are gaining in popularity and in use because they have this incredible power. So some of the advantages of using bacteriophages against bacterial pathogens is that they, when you take an antibiotic, sometimes that will have negative effects on all of the bacteria in your system, which is why sometimes we get side effects from that involve losing some of the beneficial bacteria in our guts when we take antibiotics. But a bacteriophage is really specific, so it only infects the cells that it can specifically recognize. And in the literature so far, those are generally going to be pretty closely related to the bacterial pathogen. So it has this very narrow host range, generally. They're also incredibly numerous. So I said 10 times more numerous than bacteria, but what that means is this number like nonillion. So that's an incredibly large number that's hard to imagine. There are a lot of bacteriophages, and they are just full. The soil around us is full of these potentially useful viruses. And they're also recyclable. So once a bacteriophage is being administered, it is eventually going to get kind of turned over in the environment. So antibiotics, if we take an antibiotic, probably the estimates are that 80% of them end up in going through our bodies and ending up in the groundwater and in the soil, whereas bacteriophages are going to be turned over. But these can also be disadvantages, right? So that they're very specific means that we need to find exactly the right phages for the pathogen that we're interested in. And that can be a challenge, especially when you're up against a medical condition, for example, and you need that tailored bacteriophage. They're highly numerous, but we only know in terms of their DNA about 3,000 of these. So these have been discovered and sequenced and studied. So we don't know as many bacteriophages, like we've barely scratched the surface. And because they're recyclable, they're eventually destroyed. So if you're using bacteriophages in, for example, agriculture, you might need to reapply them. They will not stick around forever. So if we think that bacteriophages are something that can be useful, then we need to find the bacteriophages that will kill the pathogens that we're interested in destroying. And that's why there are like these assassins or these ninjas beneath our feet. So we got interested about 3 years ago in trying to investigate the degree to which we could solve the problem of American foul brood using bacteriophages. And of course, American foul brood is a disease of honeybee larvae that's caused by a bacterium called Pennibacillus larvae. And you're all familiar with the symptoms of Pennibacillus larvae and American foul brood. So there's the classic roping out and the larval tongue and coffee colored larvae. These are all signs that you might have American foul brood. This is what the microorganism actually looks like in our laboratory. And of course, the spores are one of the big problems in American foul brood, in that the spores have been documented to last for over 30 years in the soil. So we can grow the Pennybacillus larvae now in our laboratory, and we can also turn the Pennybacillus larvae into spores and back. And that's important for the kind of work that we're doing. So the reason that we think that AFB is something that can be handled by bacteriophages is that in 2017, there was a report in the Journal of Invertebrate Pathology. And actually this was, I'm going to just briefly describe one experiment, but there was a lot of really compelling evidence that if you put the bacteriophages that can infect Pennybacillus larvae on a hive, that they can protect against that hive becoming infected. And so there was a natural experiment that was conducted where a single hive in an apiary of 11 hives was infected with AFB. And of course, in the United States, you're allowed to treat. This was taking place in the United States. So they were considering treating with antibiotics, and they decided instead to try treating half of those hives that had not been infected with AFB with these bacteriophages, and leaving the other half of the hives without bacteriophages. So they just subjected them to a sugar treatment. And what they saw 4 weeks later was that the hives that had not been infected with, had not been treated with the bacteriophages, most of those hives now had the AFB infection and were not protected. But the bacteriophages appeared to be guarding against the AFB spreading to those hives. And if they did the AFB Hitchcock scores, they saw that the mock treatment where they had received the sugar water, they had these high degrees of infection severity, whereas phage-treated hives didn't have any infection at all. So this is really a good sign that bacteriophages can be used to protect hives from getting AFB. So based on our interest in the apiculture industry and the evidence coming from the United States that this is a reasonable solution to AFB, we started the ABATE project, and that stands for Active Bacteriophages for AFB Eradication. And I recruited 2 students, Danielle Koch, who did most of the work that I'll be talking about today. She's been isolating the AFB causing organism, Pennybacillus larvae, and discovering bacteriophages. And today I'll talk about the most recent work that she's done kind of in the last year, getting the sequences of these bacteriophages and testing the phage cocktails. The other student is Joe Turnbull, who was an excellent master's student, who has been developing an alternative host and also working on evolving phages. So the first problem, of course, for the 3 of us is that we're not beekeepers. So we don't have access to beehives, and maybe some of you participated in this project. So we have some beehives pictured here that are on the Massey University campus, but we started receiving soil samples and bee debris samples from beekeepers, but we weren't finding phages in the first instance. So Danielle, it's kind of it's almost a joke in bacteriophage hunting that everybody has to go to the wastewater treatment plant at some point and take some samples and try to find phages in wastewater. We didn't end up finding or even managing to process those samples because of our biosecurity issues. But what we did do in order to get access to samples was we asked you. So we received over 400 samples from across New Zealand of soil and bee debris from near hives. And we did that using this infographic that kind of made this call for, please send us these soil samples. And we had the cooperation of some of the other MPI-driven projects. And of course, we're working with AFB PMP, and we're working with the monitors of AFB. So from these 400 samples, we ended up finding 30 unique bacteriophages. And in order to do that, we brought soil or bee debris into the laboratory and filtered it for everything but the very smallest particles. And we plated the Pennybacillus larvae on bacterial agar plates like this, and filtered those particles, so the very smallest particles came through and looked for evidence of bacteriophages. And these clearings in this lawn of bacteria are called plaques. And each one of these is evidence of a single bacteriophage landing on the plate, starting to replicate inside those bacterial cells, and then continuing to kind of amplify and amplify and amplify. And we have this exponential growth of bacteriophages in that little spot that eventually makes this clearing. So this is how we find the bacteriophages. And this is Danielle looking at plaques on a plate. And these are some of the phages that we discovered. So every time that a beekeeper helped us to find a bacteriophage, and we found a positive sample in one of their soil samples, we found a phage. They had the opportunity to name that phage. So phage Ali and AJG77 are both examples of names that have come from beekeepers. And I'll show you the complete list of names, but this is what the bacteriophages look like. So you can see that they have this, what we call prolate head and a long flexible tail. And this is, these are classified as Cyphoviridae. So that's the type of bacteriophage that these are. And that's the kind of distinctive look that we have to most of these bacteriophages that we found in this project. And we're still getting more images of some of the other bacteriophages, but this is just a representative sample. So that's a little bit about phage discovery, but there's a lot of work that has to happen after phage discovery in order to demonstrate both that the bacteriophages are effective and to understand some of their biology, and then eventually to get to this later stage of the project, which is testing the bacteriophages in actual larvae. So I'm going to talk a little bit about these two different streams of work that we've done. And I'll start with the host range, because you really need to establish the host range on the range of bacterial pathogens in order to start figuring out which bacteriophages you might want to include in a cocktail. All right, so this is an example of the 30 bacteriophages that we found in the project, and they have been color-coded. These are color-coded to the part of New Zealand that they're from. So we can see, you know, we're here in Auckland. So these kinds of light peachy bacteriophages were all discovered here in Auckland. And we named some of these bacteriophages when we didn't get names from beekeepers. But some of my favorite names, I think, are probably Bloomfield and Stretch. And we named a bacteriophage after Barry Foster. And we have also a Jacinda, who is from the Lower South Island. So this is an example of the host range of the full 5 bacteriophage, 5 bacterial isolates that we have from the North Island, and all of our bacteriophages. And what you can see here is that each gray square is where the bacteriophages are killing that bacterial strain. So these are 3 strains that are killed by any of the bacteriophages that we've discovered. And that's great, because that means that if that particular strain of AFB showed up in a hive, any bacteriophage that we exposed it to would kill it. Slightly more difficult are these 2 strains here on the right, 2006 and the 7957. These are only killed by Callan, Dash, and Lilo. And we do these host range tests by looking for plaques on plates. All right, so that's the North Island. We only have 5 bacterial isolates from the North Island at present. We have far more isolates from the South Island. And I have to mention the AP Wellbeing Project, but from MPI, has been incredibly important to us in allowing us to get our hands on 22 more bacterial isolates than we had previously. So Danielle had found 8 different bacterial strains in infected hive material. And these bacteriophages, I should also mention, have all come from healthy hives. So that's an indication that the presence of bacteriophages is supporting hive health. All right, so we have lots of strains from the South Island, and we can see that we have more strains that are only infected by Callan, Dash, and Lilo. We have lots of phages, lots of strains that are infected by many of the phages, especially these that are listed here at the top. These are great phages. They kill a lot of these bacterial strains. And what we discovered was that in the lower South Island, there were 2 bacterial strains for which we have not found any bacteriophages yet. Yes, so all of these bacterial isolates that are listed along the top are, we don't know that much about all of them, but they're all Penibacillus larvae variants that have come from different samples of infected hives around New Zealand. So this gives us a little bit of a sense of how variable the Penibacillus larvae pathogen is in New Zealand. And what we want is ideally to put together a mixture of bacteriophages that would be able to kill one of these strains, no matter what strain showed up in your hive. And so one problem that we have right now is that we have 2 isolates, both from the lower South Island, for which we don't have any bacteriophages yet. And that's something that we're going to want to fix in the future. All right, so to summarize what we've learned about the host range, so far we have 33, we thought 33 Penibacillus, but it turns out that a couple of those were the same once we sequenced them. So we really have 30, I should correct that. And each one of these 30 bacteriophages is able to destroy between 53% and 87% of the Penibacillus larvae isolates that we see in New Zealand. We can see that we have this uneven distribution of Penibacillus strains, so we should probably get some more strains from the North Island, and we're in the process of doing that. And we've also found these 2 strains that are not able to be infected by any of the bacteriophages that we've discovered to date. Okay, so that takes us here. And we really wanted to do some DNA sequencing. And part of the reason for that is to understand how different these bacteriophages are from one another. Part of the reason for that is to see if we can see what types of infection they do. And part of the reason for that is that bacteriophages can sometimes conduct a life cycle where they contribute genetically to the bacterial strains. And so what that means is that the, sorry, I'm reading the question. So what that means is that sometimes we have genes inside the bacteriophages that are going to change some of the biology of the bacterial strains. And we might want to avoid some of those. So that's part of the reason that we do this genome sequencing. So I'm going to talk about this stream of work first, and then I'll talk about this lower stream. So we wanted to do the DNA sequencing. And so I have a question, why is it you can treat AFP in some countries but not others? And I think that the answer to the question might be the law. So in New Zealand, we're not permitted to treat AFB with antibiotics because the industry has decided that we don't want to encourage antibiotic resistance, and we don't want to introduce those types of chemicals into our hives and into our honey. But in the United States, you're allowed to treat with antibiotics. Okay, so in order to get the DNA out of our bacteriophages and send them to a genome sequencer and get the DNA sequence out, we have to have a high titer or a high concentration of those bacteriophages. And in this project, we were finding we could discover these bacteriophages, but they were in low concentration. So we could do the things that we would normally do with bacteriophages, and they were maybe 1,000 per milliliter. And what we really want is like 1 billion per milliliter. So we want much higher concentrations, both to use them practically and also to isolate their DNA and study it. So we needed to solve this problem. And we found this really interesting paper that was published in 2019 that talked about something that was called the modified Appelman's protocol. And this is something that they had been using in Poland to do like an experimental evolution of these bacteriophages to get them to swap bits of DNA between them. And that had the property of expanding the host range if you had the right bacteriophages. And so we looked at this and just thought, well, I mean, we could change this a little bit. And instead of mixing all of the bacteriophages after they've done lysis in a 96-well plate, we could just take this group of bacteriophages that grew in these different dilutions and put them back on the same bacterial host again, and just keep doing that again and again. And that might evolve the bacteriophages to be better at infecting that host. And if they're better at infecting that host, then maybe they'll be better at increasing their concentration. So the Appelman's protocol is normally done for about 30 days, but we saw that in as little as 4 days of doing this repeated infection in these 96 well plates, we could find huge increases in the concentration of bacteriophages. So we had, these are just some examples of some of the bacteriophages. They were maybe 1,000 per mil, maybe 10,000 per milliliter. And then after 4 days of evolution, we could get much higher concentrations, so millions per milliliter. So 10,000-fold improvement in the concentration in as little as 4 days. This for us was just unbelievable. So we were really happy to find this kind of improvement and such a simple protocol that just took 4 days in the laboratory of propagation. So this was really a game changer for us, and it allowed us to extract the DNA and start sequencing these bacteriophages, which changes the game for us. So we managed to do some experimental evolution, which is going to lead to small nucleotide changes that occur randomly in the bacteriophages, and then select for the ones that are improving their ability to infect. So that was great. So next we wanted to take the information about host range and also what we were learning from DNA sequencing and design some bacteriophage cocktails. And again, the idea here is to find a set of bacteriophages, that would kill most of the bacterial strains that we might find in a hive. So Karen asks, the principle of survival of the fittest and perpetuating the strongest lines. That's exactly right. So we were allowing evolution to take place over the course of 4 days in these simple little 96 well plate liquid experiments. So yeah, that's every time anything replicates its DNA, it has chances of mutation. And what we were doing with the experimental evolution is just allowing the mutants that were a little bit better to win out in this experiment. And that's something that we do in my laboratory a lot. So it's kind of fun for me to bring these two halves of my interests, evolution and bacteriophages, into the same project to solve a problem that we were having. Okay, so let's talk a little bit about cocktail design. There are lots of ways you can imagine taking this set of bacteriophages and coming up with a small number of them that you would put into a mixture, and then that would be the thing that you would want to put on hives to protect them. And what we eventually ended up doing was deciding that if we had any of these strains of Penibacillus larvae, any of these bacteriophages would do. But if we choose the ones kind of higher up in this list, they also have the chance of killing possibly these green strains. Some of them, if you picked them, would also kill the red strains, and some of them, if you killed them, would also kill the purple strains. So we've listed them in this order so that they have these qualities. But we thought, well, we'll make sure we have something from this blue group and something from this green group, something red and something purple. And of course, in the future, we'd also like to add something that would kill these outliers that we don't have any phages for at the moment. So and then there was one more piece of information. So the project that Joe, the master student, had been working on allowed us to see the genome for Lilo before we could see the genome for anything else. And of course, in this purple group, this whole set of strains, including strains from the South Island and the North Island, have to have either Callan, Dash, or Lilo in the mix in order for those to be infected, for those strains to be infected. So that gave us some important information, and that was that Lilo was probably not going to be a good choice for our bacteriophage cocktails. So we came up with 4 different cocktails to try. Callan, Logan, Matzi, and Freya were in cocktail 1, and you could see the names of the other ones, and you could see these kinds of color codes that describe which sets of strains they can infect. And we wanted to try all 4 of these against some small number of bacterial strains that are the examples of what you might find in a hive. So the way that we did this was, again, to use 96 well plates and to just set up a serial dilution of the bacterial strain of interest. So here are the bacteria, and also serial dilutions of the bacteriophages. And we had, all of these wells had either the bacteria or the bacteriophages or both. And so in this area, you can see the results of having both. And then we just, after we did the infection, we let that go for 24 hours or 48 hours. And then we took a tiny amount of that liquid, and the white bits on this plate represent single cells that had survived that infection. And in this red box, these are the numbers based on the literature that you would see probably of bacteriophages, around 10 to the 5 you could get into a hive. And this is the dilution of the bacteria that you would generally see in a hive. So that's the box that we really kind of want to use as the guide to see whether or not this is working. We want to see as much cell death or as few bacteria growing here as possible. So this is an example of 2017. And here we're infecting with cocktail 1. And if we expand that, we can see cocktail 2 and 3 and 4. And what should be fairly obvious here is that cocktail 2 and 4 appear to be performing a bit better than cocktail 1 and 3 in terms of clearing the bacteria on this plate. And cocktail 2 and cocktail 4 seem to be doing really well. Ooh, Karen is asking me about viral reassortment, and that's something that we would like to look into more, using that Appelman's protocol. So using that experimental evolution approach, we hope to improve some of these phages further by doing recombination in the future. But that's a great question, and one that's very close to my heart. So we tried these different cocktails out on 4 different strains that were kind of representative. So here's the 2006 strain that we find here in the North Island. And you can see again that cocktail 2 and cocktail 4, with a little bit of contamination, it looks like in our experiment, are really doing an excellent job of killing this Penibacillus 2006 strain. So again, cocktail 2 and cocktail 4 perform better than cocktails 1 and 3. And then 8100 is a South Island strain. And again, in the area that really matters, we're seeing pretty good results with cocktails 2 and 4. And of course, P1627 is one of those 2 strains from the South Island for which we don't have any bacteriophages. And we wanted to try this just to see, like, if we put these bacteriophages in combination on this strain, and we don't think that any of them are going to be able to infect it, do we see any kind of emergent property of this mixture suddenly doing a pretty good job of killing? There are some biology reasons that that kind of thing might happen, but actually we don't see that in this case. So it really looks like we're going to have to find these bacteriophages to kill P1627 and the other South Island strain. So about at this point in doing our cocktail testing, we got back some of the genome sequences for the first 6 bacteriophages that we had put through that Appelman's protocol and then annotated. And so what you can see here is this set of 6. It includes Lilo and Dash and Callan, Abate and Zed, AJG77 and Loken, and these purple bands between each of the phage genomes represent areas of high similarity. So Logan and AJG77 are incredibly similar to one another, but a little bit different, and so on and so forth. And what we were kind of devastated to see is that our phage cocktail that was doing so well, phage cocktail 2 and phage cocktail 4, both have Dash. And unfortunately, Dash has this same honeybee toxin, this larvin, that kills honeybee larvae that Lilo has. And so we wanted to see if, in order to avoid putting together a phage cocktail that has this honeybee toxin in it, and increasing the numbers of copies of that gene in the environment, if we could replace the Dash phage with the Callan phage in cocktails 2 and 4. And unfortunately, as soon as we replaced and took Callan out, as soon as we put Callan in and replaced Dash so that we wouldn't be adding more of this larvin, we saw much poorer results. And we're not exactly sure why that is right now, but to Karen's question, we would love to do some more recombination experiments with these phages and see if we can get the balance of qualities right, where we have Callan, which doesn't have the phage, the larvin, this honeybee toxin in it, but does have the qualities that make it so good at killing the bacteria in cocktails 2 and 4. So using these types of experimental evolution approaches are ways that we can hopefully get the right combination of traits by doing recombination with these bacteriophages. So that's a great question. All right, so we have sequenced 8 of the NZ bacterial isolates. And I know that the AP Wellbeing Project is in the process of sequencing the rest. And I can say that all of those Pennybacillus strains have this larvin toxin in their genomes, but I still don't like the idea of making a phage cocktail that has a toxin in it and exposing honeybees to something like that. So this is an ongoing issue for the project that we are hoping to resolve, and we would like to do more work on this in order to figure out if there's a way that we can get the balance right in the traits. So the 2 work streams that I've talked about were the experimental evolution and the DNA sequencing. I hope now that you can see why the DNA sequencing is so critical, so that you know what the traits are of the viruses that you're intending to use to protect a hive. We've gone through some cocktail design and some in vitro cocktail testing where we tested them in 96 well plates, but you never really know from a 96 well plate experiment what it's actually going to be like for a honeybee larvae to be exposed to these bacteriophages. So we have some wonderful colleagues, Ashley Mortensen and her team over at Plant and Food Research. And this summer, since now kind of spring is passing us by and we're still in lockdown here in Auckland, we're going to be sending Danielle Koch to the Plant and Food Research facility, and she's going to be rearing honeybee larvae in 48 well plastic plates. And that's going to give us the opportunity to expose them to the bacterial pathogen, and to, in advance of exposing them to the bacterial pathogen, to give them the bacteriophage cocktails and see how well do the larvae respond to the presence of the bacteriophages alone, the bacteriophages, and then the AFB pathogen, or the AFB pathogen alone. So we're really excited about doing those experiments because we think it's really going to help us to understand what the bacteriophage cocktails will be like in a live hive. And of course, we're hoping that experiment will happen in January now that we're hopefully not going to be in lockdown forever. So that's still to come. Okay, so the summary of the latter half of the talk is that we've identified some effective cocktails for most New Zealand strains of Pennybacillus larvae. So we think that we are pretty close to being at the point where we could say, here's a bacteriophage cocktail. If you put this on your hive will be healthier and safer than it would have been otherwise. Of course, the conundrum right now is what to do about this toxin, and can we eliminate this toxin from these bacteriophages and come up with the perfect mixture of bacteriophages. So moving forward, we are going to be testing these cocktails and trying to eliminate the toxins that we find in some of them. We'd like to isolate more bacterial strains from the North Island, and we'd also like to find bacteriophages that will be able to infect the 2 strains from the South Island for which we don't have any bacteriophages yet. And ultimately, we are going to be partnering with groups like Comvita and some of the apiculture training courses to perform blind field trials, where we will either apply a phage cocktail or a sugar solution and look at how well the bacteriophages protects those hives against AFB. And of course, I'm in the process of finding more funding to continue the project, but we have been funded by both AgMart for the next phase of the project for the next 1.5 years, and also the Honey Trust. And we're incredibly grateful for both of those sources of recent funding. All right, so in wrapping up, and then I can take some more questions. I really think that bacteriophages are incredibly promising for solving problems in agriculture. And I would love it if 10 years from now, we're not only solving problems in agriculture with bacteriophages, but we have grown our intellectual and physical infrastructure to the point where we might be able to solve medical problems with bacteriophages. As we see more antibiotic resistance evolving in bacterial pathogens around the world, I think that this is a really promising source of natural solutions to the problem of bacterial pathogens. And so with that, I have to say, you know, I've been at Massey University for 10 years and it's been great. I am now moving to the University of Canterbury in basically in a month. So by January, me and my laboratory will be at the University of Canterbury, but we will still be thinking of our friends here in Auckland and working to find solutions for your beehives as well. And I have to say, you know, AgMart, the Sustainable Farming Fund, have contributed to this project in the past, and the Honey Trust and also AgMart again are contributing to our future for this project. We're grateful to HHMI for the training and for the science education platform that they've allowed us to be a part of at Massey University. And while I love bacteriophages and I am excited about this work, we also do other types of projects that involve bacterial evolution, and that work has been funded by the Marsden Fund. And with that, I should be able to take some of these many, many questions. So I think I will stop sharing for the time being, and let's have a chat. Thanks.
There are several questions there on the list. Do you want to simply pick up on those and answer them, Heather? You comfortable going through those?
Yes, it should be good. Hopefully I understand them all. And if I don't, I'll have to get some additional comments. So Peter says, can you say more about larvae? What is the advantage for a phage carrying the larvae gene? So one of the things that I'm not talking a lot about right now is the fact that bacteriophages also sometimes have another life cycle where they integrate into the bacterial chromosome and just hang out for a while. And you can imagine that, especially in a situation where there aren't a lot of hosts, if there aren't a lot of bacteria to infect, just sitting and replicating along with your bacterial strain can be a really successful way of living. So bacteriophages will shift between these two life cycles with some frequency. And so for a bacteriophage, if it has a larvin gene in it, that might make the bacteria that it sits in slightly more pathogenic, and that might be good for the bacteriophage once it's integrated into the bacterial chromosome. So that's why they have the larvins. Bacteriophages, more than anything else, are a little selfish genetic element that carry with them tools that make them better at replicating. And so that's the story on the larvin. And but we can propagate bacteriophages that don't have the larvin because we're the ones that are choosing what traits they have or don't have. It's going to be really problematic for us to get into genetically modifying bacteriophages for a lot of reasons, which is why we really like the idea of using these recombination experiments and trying to tip the scales of natural selection towards choosing bacteriophages that don't have the larvin. And one way that we might be able to do that in this case is by choosing bacteriophages that have less DNA. So they might be able to replicate faster or in higher numbers if they don't carry that larvin in this kind of artificial setting. So those are the types of tools and tricks that we'll be using in the future to try to get away from bacteriophages that have larvin genes. Okay, so Karen asks, are the cocktail solutions applied as an aerosol to cover surfaces or feed the bees to ingest and spread? So this is a great question. Thanks, Karen. There are lots of ways that you can imagine administering bacteriophages. In some instances, and we haven't started to get into that side of the project. In fact, I'm applying for some IMBY funding, hoping that we can start investigating how do you apply the bacteriophages that we have How stable are they? How long do they hang around? Those kinds of questions. So you could put them in a liquid solution. So you could put them in like a water trough nearby, or you could potentially do something called lyophilization, where you make them basically into a dust, and then you can dust. The key is going in whatever administration technique you use. And some people have used sugar water, but I can see why people might want to steer clear from sugar water if you're really trying to get honey that has these native or non-native plant characteristics. So there are lots of methods that we can use, but the trick is going to be you've got to get the nurse bees administering the bacteriophages to the larvae. And so that's really the critical point in making sure that those bacteriophages are in the place that they need to be in order to protect the health of the larvae. So there are lots of ways that we will be trying that. I know people have tried smoking the bacteriophages into hives, or tried water solutions and sugar. Okay, another question from Karen. How do the phages survive if there are no host bacterial targets available? Yeah, so with these bacteriophages, we don't know right now if they are able to infect anything else in the environment, and that's something that we could investigate further in the future. And that's part of the reason that if the bacteriophages are present and the pathogen shows up, then they can amplify on it, and then we'll have more of them. But that is exactly why we need to test how long do the bacteriophages hang around if there isn't any AFB present in the hive. And that's something that that the INBI project that we're trying to get funded would also investigate that question. So that's something that we would like to know as well. Is the toxin a byproduct of the bacteriophage life process, or is the bacteriophage DNA directly toxic to the larvae? So a bacteriophage, as it's kind of floating around as a little protein particle, doesn't have any energy, doesn't have any ability to replicate itself, doesn't have any ability to produce toxin or excrete anything. Like it is actually a matter of debate. Here, I'll got my little hot pink bacteriophage here on my desk. It's a matter of debate as to whether or not a bacteriophage is alive, right? Like, so it can't do anything until it infects a cell, and then it can only produce proteins and make copies of its DNA and do all the things it needs to do once it's using the energy of a cell. So, so a bacteriophage itself, if it has the larvae in its genome, it's not doing anything. So it's not, the larvae isn't a problem until that bacteriophage integrates into the bacterial chromosome and is sitting there and is being expressed by the bacterial cell itself. And that's when the larvae becomes a problem. And what I would hate, because I've studied this process of horizontal gene transfer where bacteria, partly through bacteriophages, exchange genetic material. What I would hate is to see us use a bacteriophage that has a larvae in it, and that larvae start ending up in more bacterial strains than it's in right now, and increasing the number of things that are pathogenic towards honeybees. So that's why I think sequencing is so important, and really knowing as much as possible about the biology of these bacteriophages before we start using them is absolutely crucial. And that's a discussion I'm having with some of my feistier colleagues at the moment. So that's an ongoing battle. All right, Tim asks, does the presence of bacteriophages in the hive account for the antibiotic properties of honey? And if so, are these phages properties influenced by honey types such as Manuka? So there's a lot in that question. The antibiotic properties of honey is especially of Manuka honey, has been studied, and it's down to a chemical that I'm sure somebody in the chat remembers exactly the name of the chemical right offhand, but I don't. So it's a combination of the work of the honeybees and the Manuka plant itself to produce that little chemical, and that's what actually makes honey have antibiotic properties. So it has antibiotic properties against like Staphylococcus and E. coli, and especially in bandages and things like that. So it's not because of the bacteriophages. If so, these phages properties influenced by honey types such as Manuka. One question I would love to know, and I'd like to get together with some of my other colleagues that work on the microbiomes of floral plants is, where are AFB in the environment? So where is the Penibacillus larvae hanging out in the environment so that it gets into the beehive in the first place? And it may be that the bacteriophages that fight against AFB are present in things like Manuka. It may be that the honeybee pathogens are hanging out in things like Manuka. We just don't know where the natural reservoirs of these pathogens are, and I would love to know that. But right now, I don't have any idea. So I don't know where the bacteriophages are hanging out, except that in New Zealand, every bacteriophage that we have isolated from hive material and from soil have only come from healthy hives. And we have sampled a lot of unhealthy hives, and we have not found any bacteriophages in them. So I think that that's really an interesting kind of factor. Certainly, I can say that in the United States, they did find some bacteriophages against AFB in AFB infected hives, but that's not been the case so far here in New Zealand. Okay, is the aim prevention or treatment in hives? Jackie, that's a very tricky question. So we are, of course, in New Zealand, not permitted to treat hives that are infected with AFB. Any hive that's infected with AFB has to be doused with petrol, and it has to be burned within 7 days of discovery. So because those are the regulations here in New Zealand, we would never suggest that anyone should try taking a bacteriophage cocktail to treat a hive because it's not legal. But what we can do is we can put these bacteriophages in the hives in order to protect them against getting AFB in the first place. And in the natural experiment that they did in the United States, of course, they saw that if a single hive had been infected and you protected the other hives in the area, then you were less likely to see the spread of that AFB infection. And without getting into how AFB gets between different hives, I think that that's the idea that we could protect, especially if we think, maybe a neighbor has AFB, or if we're in one of those hotspots that the AFB PMP has reported for the country, or if we're starting to get the idea from some of these reporter apps that we might be at risk, that would be a great time to start protecting with a bacteriophage cocktail. All right, Peter's got a question. Would it be possible to synthesize a minimal phage to bypass the genetic modification problem? I think most people, that's a good question, Peter. I think most people are going to consider, genetic modification in New Zealand has it's very loaded. And I think in an industry where we are known for this kind of pure natural product, if I did find some way of synthesizing the DNA and then getting a little tiny bacteriophage made that didn't have any negative properties, I think people in the industry would probably be very wary of that. But if I can say, I have taken these bacteriophages from healthy hives, I have naturally evolved them to not have any of the negative properties, and I have tested them, I think that that's something that, in my impression, is that more people will be able to get behind that kind of a natural protective product than something like a synthesized or genetically modified bacteriophage.
Thanks very much indeed, Heather. I think you're probably quite right. Adding genetic modification into what is supposed to be a healthy natural product is almost a contradiction in terms. To go back to, I think, one of the first questions about why we don't treat in New Zealand, I found a very early account of beekeeping in New Zealand, 1870s through to 1914, in the club's archives when I was doing a cleanup. And there are references in there to AFB having arrived in the country and being found, and actually having infected the main hives at Ruakura, the research centre and so forth. And of course, this occurring 1914 or prior to 1914 is well before antibiotics had been discovered. So I think simply the regulation came out at the time in New Zealand. There wasn't a way of treating with antibiotics against AFB, and therefore the regulation was introduced to say you burn the hive, and the Americans subsequently amended to say, oh yeah, we can treat with antibiotics, and New Zealand has simply maintained its previous standard of saying, no, we'll just destroy the hive, and that's the way we go with it. So I think that might have been one of the contributing factors to the way in which we approach it and how we're different to the rest of some of other countries.
I think we're doing ourselves a favor. I think we're doing ourselves a favor by not using antibiotics, because that will naturally select for antibiotic resistance, and that's a whole heap of trouble.
Yeah, indeed. That was fascinating, both for what you're trying to do, the progress that you've made, but also for the stages that has to go through. I don't think any of us looking from outside, unless we have that sort of laboratory or research background, have an understanding of the difficulties that are involved in and the steps that you have to go through between having a good idea at the beginning and then coming up with a product at the far end. So I was fascinated to actually look and see what you're doing, how you're doing it, what you have to tackle, and all the different steps to it. Thank you very, very much indeed on behalf of all of us sitting in and attending and listening. And we wish you the very best, both in your personal, you'll move down to Christchurch in Canterbury. It's a lovely city. Having been born there, but I'm biased. But also all the best for your work in the future, and our best wishes that you find a solution for a product that I know has, or a solution for a problem that I know has devastated many beekeepers over the years, who have worked hard in their hives through no fault of their own, have found to have AFB in their hives, and just the devastation of having to pour petrol through your hive, burn your girls that you've worked so hard to keep. If we can find a better solution for it, then you will have the eternal gratitude of both this club and beekeepers all across the country and elsewhere as well. Thank you very much indeed for today.
Thanks, guys. It was a real pleasure. And if you have more questions for me, you can find me online. And it's always a pleasure to hear from the beekeeping community. It's really been fun to learn more about all of you, and you had great questions. So thanks a lot.
Thanks, Heather. Enjoy the chocolates.
I'm really delighted. Thank you so much.